Hi, everyone, and welcome to this edition of Budding Solutions, a series on disease and pest management in cannabis sativa. My name is Kim Leonberger, and I'm an Extension Associate in the Plant Pathology Department at the University of Kentucky. In this episode, we are going to take a closer look at mites. To discuss this important issue, I would like to introduce Dr. Raul Villanueva. Raul obtained a Master's of Science from Queen's University and a PhD from the University of Florida. He is currently an associate professor and extension specialist of field crops in the Department of Entomology at the University of Kentucky. As the sole faculty member of the Department of Entomology based outside of the Lexington campus, he provides entomological support throughout Western Kentucky. Raul works on arthropod pests of hemp, including aphids, corn earworms, and russet mites. He also conducts research and extension efforts towards pests affecting soybeans, corn, and winter wheat. Raul actively promotes integrated pest management practices, the judicious use of insecticides, and the conservation and enhancement of natural enemies. Raul is going to share a few slides with us about mites, and then we'll have time for a few questions. Welcome, Raul.
Today I'm going to talk about tallies of hemp russet mites through photographies and the assessment of the efficacy of conventional and organic insecticides. And for this, I have co-authors Armando Falcon-Brindis, Zenaida Viloria, and Christine Bradley. The hemp russet mite is a very interesting mite that affects and feeds on plants, on hemp plants. In this group, one particular issue is that they have two pair of legs, but also in addition to that, they have only two nymphaline stars compared to other mites. And the adults can reach to 180 microns there, so they are very tiny. And also, the reproduction is via spermatophores, so the females lay an fertilized egg on the plant, and then the male fertilizes it, and that's how this reproduction is done. So another important factor is that due to the small size, it's very hard to notice on the plants. So you start to notice when you have the symptoms. But for disperse, they have a particular behavior where they go to the edge of the plant or the leaves, as you can see in the picture, and they use the caudal hairs on the bottom of the body, and then raise the body and then extend their arms. So, and then they are carried by the wind. Also, in addition to that, in many areas of the USA, this dispersal was done through the trade of the clones. The symptoms for the presence of infestation of these russet mites are folding of the edges, as you can see here, and this folding of the edges can be upwards, as you see in this image, or it can be downwards. So in some cases, the description is that the folding is upwards, but it's in both directions. Also, you can see when there is high populations that these mites concentrate on the new growth of the plant. In addition, if infestation occurs in seedlings, it can stunt or kill plants. As you can see here in this study, we transferred hemp russet mites at 5 days old, and then you can see at 23 days old, all the leaves are folded. When you see further infestations, so further age of the plant, you can see, compare the control plants versus the infested plants, you can see a difference on height. So this affects the growth of the hemp plants, especially when there are high numbers of them. So one aspect that we try to evaluate here is how to tally RFI mites, because they are very hard to count with the naked eye. So we need to use high magnification glasses. And prior to this study, I worked with these hemp russet mites on citrus. And you can see the damage that they cause on citrus. This fruit actually is not for consuming on the table, but for use around that time, maybe 20 years ago. So counting mites can be done with microscopes, with magnification lenses, and also with mite brushing machines. However, All these methods are tedious, time-consuming, and need to be done in a relatively short time because the mites that are on the leaves can move out if they are not counted probably 1 day, 2 days. And that's kind of an issue with this study. In addition, also we have that when you use the mite brushing machine, the leaves of the hemp plants are very fragile compared to citrus or even to tomatoes. But also we have the situation that we have a composite leaf, and in this case we decide to use only the middle biggest leaflet to do the counts. And for that we use a simplified tally process where we use a handheld microscope. But to evaluate that, we did a comparison of tallying using handheld microscopes versus stereo microscopes. So here are the results based on the count of Hemp Russet mites using microscope and the Hemp Russet mites using photographies. You can see in both cases the control was kind of in the middle of the population of these trials in both cases. The highest numbers were found on the seal matrix product in both cases. The lowest was in BoteGHA in both cases. And so this is kind of a proof that we can use that one. Furthermore, we did other studies where we compared the linear correlation between the log of the stereoscope and log of the image-based tallies, so using photographies. And under two experimental conditions. One was in the field and the laboratory, and you can see that in the field, here the gray area, there is a positive correlation. On the laboratory, the same situation. We have a positive correlation using these two ways. Also, we did a pairwise correlation of laboratory and grown hemp samples, laboratory and field grown hemp samples. And you can see we use the distal, the middle, and the proximal areas where we took pictures. And the highest numbers or the highest correlation coefficient was in the proximal area. So after conducting these studies, we decided to use the proximal leaf area, and we base our studies on these counts. So we did efficacy of pesticides in 2021 and 2022. And for this, we use plots that have 12 plants per plot with 3 rows each, but we use only the middle plot, as you can see here. And in this middle plot, there were 4 plants. So we removed some leaves from these middle rows after the sprays or before the spray. And these are the results for 2021 using conventional acaricides, biological-based, and mineral products. So you can see here the most effective products were Fujimite, here on yellow, ABBA Ultra in light blue, and also we have here BoteGHA. This is a biological-based insecticide. And later on the study, we found that also BW281, this is a mineral oil, was effective. However, you can see that Stifle, that's a conventional insecticide, was not very effective, and also other products. So they were not significantly different than the control. So in this case, we did 3 applications, as you can see here. The first was in August 6, all treatments were sprayed. The second was all treatments were sprayed except the ABBA Ultra, Fujimite, and Stifle. Those are the conventional insecticides. And the third was done with all treatments, but except the Fujimite and Stifle in the control. Okay. In 2022, we have here other products, but in this case, we also have 4 sprays. So the first one, all products were sprayed. The second one, all products were spray except portal and cannamide. And the third was all products sprayed except cannamide. And the fourth all were sprayed except portal. And control was not spraying in any of the case, just to be sure that that happened. So again, in this year, ABBA Ultra, SuffOil, and also Portal were very effective. In this case, BoteGHA, that was very effective in 2022, was not. It didn't have such effectivity. Okay. Now, the main findings of this study are presented here. In 2021, Fujimite and ABBA Ultra, these are conventional insecticides, were very effective, as well as BoteGHA. BoteGHA is a biological-based product, and in 2022, this was not very effective compared to 2021, and maybe due to environmental conditions that I'm going to describe below. In 2022, the most effective were Portal, ABBA Ultra, as well as SuffOil. Cannamide, another conventional product, was not significantly different from the control. Now, I believe that the organic pesticides were affected by environmental conditions in 2022. As you can see this graph, it shows that during the plant growth development and the Hemp Russet establishment, the temperatures were 6 degrees Fahrenheit. The relative humidity was 10% points below, and the accumulated rainfall was low in 2022 compared to 2021. So I believe this probably caused the reduction efficacy, especially of BoteGHA. Okay. Also, in addition to that, the use of organic insecticides need to be repeated. The effectiveness of these sprays were not observed immediately, but after the second spray, and the most effective herbicide mites was after the third spray, especially in 2022. Phytotoxicity was not observed for any of these products later in the season. There were some plants that have some of these spots, but they were not really of such importance. So this was studies were funded by the USDA Agricultural Research Service and the IR4 funds, and we have also appreciate the help of all the people that work in Princeton, the University of Kentucky Research and Education Center. Thank you.
Thanks so much, Raul. That was very interesting. I do have a few questions for you. With the first one being, you showed some really great images of these mites. Can you talk a little bit about how it's important for growers to understand the biology of mites and how that plays into them making management decisions?
Sure, growers need to be aware of the presence of these mites because, as I mentioned, they are very tiny. So probably they need to have a high magnification tool. Right now you can buy a very cheap kind of electric handheld magnification tools. That can go and be aware that the plants don't have these mites. Especially if they grow hemp indoors, especially when they are tiny for propagation. These mites can kill the plants, reduce the plants very rapidly because they have a very rapid development growth. Mm-hmm.
Excellent. Can you also cover a little bit about the host range? You touched a little bit on this, but how does the host range of mites really play into the importance of them as pests for hemp?
So in this case, the Hemp Russet mite is a very interesting mite because it affects only hemp. So if there's no hemp, they are not going to be reproducing. So it affects hemp that is used for for grain, for CBD, and for THC. And I believe in many cases, especially as I mentioned, when you start to grow these plants indoor, they can be very negative on the growth of the plant. So especially for CBD and THC.
Very interesting that it has just that kind of specific host range.
Mhm.
All right, last question for you. You've touched a lot on some of these biologicals, but could you talk about how the efficacy of things like biologicals and maybe even cultural controls compare to some of the conventional pesticides that you might use, which we kind of have limited access to?
Sure, the biologicals are very important, but their effect on controlling or reducing populations of pests are not as fast as conventional insecticides. So one thing that you need to do is probably use sometimes in preventive ways. So probably spray before the pests are present, and or when the pests are very low numbers. Also, you need to do probably repeated application, maybe 3 or 4 times, because their efficacy is affected by the environmental conditions, as I'm showing here, and also the efficacy is not as long as conventional insecticides for that purpose, for that reason. And they can be killed by, for example, in this case, BoteGHA. I believe that it didn't work in 2022 because we have very low relative humidity, and also we have absence of rains and very high temperatures. So that's very important.
Thanks so much for joining us today, Raul, and sharing all this wonderful information. Just a reminder to our viewers to please check out the description below the video for additional resources. Thank you for joining us, and we hope to see you again on the next edition of Budding Solutions.